Good morning, one and all. I'm Genga, a volunteer of Kotak. We are gathered here today to celebrate the fifth anniversary of Mind. The journey which started from a small dream has now become a big dream which hundreds of people cherish. Of the five dreams Mind envisions, research is the most important one. We all by now know, thanks to all the awareness program by Mind, that muscular dystrophy and spinal muscular dystrophy are genetic disorders, and there is no particular medicine available now to cure these diseases. And if available, they are really very costly. It is only through gene therapy or modification in DNA one can cure this disease or prevent it from transferring from one generation to the other. This is where the importance of research lies in. If we are able to open more research centers in India and make medicines in our land, we will be able to cut the cost of these medicines and make it affordable for the normal people. It is with this aim in mind, Mind has started its research program Praditi, wherein we pinpoint the importance of research and also provide information about the developments in the research field. Today also, we are gathered here for a discussion which is based on a heated topic, therapies being developed for muscular dystrophy, current and future trends. We have here with us today Dr. Sudha Bhattacharya, a molecular biologist and co-founder of World Without GNA Myopathy, to take part in this panel discussion. With due respect, I welcome Dr. Sudha Bhattacharya to this program. Welcome, ma'am. I also welcome the chairman of Mind, Sri Krishna Kumar KK, to moderate this session. Welcome, Krishna Kumar sir, and I hope of everybody who have joined here today find this session informative and knowledgeable. Thank you.
First of all, thank you very much for inviting me to this meeting. Congratulations to the team at Mind. You're doing a really mind-blowing job, and my best wishes to you. So what I've been asked to do since I'm a scientist, that if I can give some sort of overview of what are the therapies which are being developed for Duchenne muscular dystrophy in particular. And of course, these will also be relevant for other myopathies and muscular dystrophies. But I'll be focusing on DMD. So I know that many of you are not very comfortable with English, but maybe next time I'll try to learn Malayalam. So, but this time we have to do with English. Now, the thing is that, for us who are patients, we are really desperate that when can a treatment come for us? That is the question foremost in all our minds. In DMD, there's huge amount of research going on all over the world. So when so much work is going on, how come we don't really have that level of therapies available which can give relief to the patients? So, there are several issues in this because to develop a new drug is quite a time consuming and also it needs a lot of care. You have to be sure that the drug is safe for human use. It's not that, it makes more problem than even before. And also that it actually does what it is supposed to do. That means to give us relief from our symptoms. So when you have to achieve this, you can't test the new drug which you are developing. You can't do it straight away on human beings. You have to use some other system. And generally, we have animal models, like we have a mouse model or dog model for DMD, which are quite good. And they are used first to test out the new drugs. And then when it is seen to be good in those models, then only it is tested on human. Now it is found quite often that even the drug has worked in mouse and dog model, it actually doesn't work in human. So that is another challenge that we have, that we don't really have a model which replicates the human situation completely. So that's why the drug development, although as I will tell you that so much work is going on all over the world, But drug development takes a lot of time because it has to be done with extreme care, and it must have the efficacy that we require. So now let's look at what is going on in the world. Uh, so before that, what I will do is that just give you a little bit introduction. Although I think most of you are already familiar, but just so that we are on the same page. Now, the diseases that we are looking at, most of them are genetic disease. That means there's a mutation or change at the DNA level itself. So, little bit we have to know that what is DNA, which I'm sure all of you know, but just to refresh that. So, here I have just a very simplified way to show a DNA strand. A DNA is a huge molecule which is composed of a string of letters like ATGC, which is repeated millions of times, literally millions. So here we are showing only 12, just for simplicity, but this is repeated many, many times. So in the dystrophin gene which is the gene where mutation happens in DMD, it's a huge DNA, which is a repeat of 4 letters, ATGC, millions of times. That so we have, it carries the code of life, as you have heard, and this code has to get translated into another code that is of a 20-letter word in protein. So DNA first gets copied into an RNA molecule, messenger RNA, and this RNA molecule then gets what we say translate into the protein sequence. So this is how the information in DNA is transferred to the protein. So in our case, this is the dystrophin gene, and this protein is the dystrophin protein. So now if there's any change in the DNA, for example, if instead in place of this A, it becomes either T, G, or C, then that change we call it a mutation. And that mutation will then change the letter over here in the protein, and that can destroy the function of this protein. So that's why the mutations which take place in the dystrophin gene, they can destroy the function of the dystrophin protein, and then it leads to the disease. So when this mRNA, messenger RNA, is being translated into protein, so it's a very precise process. It starts at a particular location, and it also has to end at a particular location. That end is called the stop. So there is a signal that, okay, now stop. But sometimes what happens that actually a mutation takes place in the DNA, which gives a wrong stop signal. It's not supposed to stop here, but it stops. So that is also a mutation found in DMD, and I will talk to you about that also. So now a little bit about the dystrophin protein itself, which is there in muscle, and it's a very important protein for muscle function. That's why in DMD, when this protein is affected, then the muscle function is very, very much severely compromised. So this protein is a huge protein, very large protein of the cell, and it's present inside the cell in such a way that one end of this protein, it binds to the cellular inside component. The other end binds to the cell membrane, the covering of the cell. So in the muscle cell, you know that the muscle contracts and relaxes. That's how the muscle does the movement. So when there's a contraction of the muscle, then the dystrophin actually shields the membrane from any damage, because the contraction creates a force and the membrane can become damaged. So dystrophin actually protects that. So that's why we find that in DMD, when dystrophin is not there, then it is quite easy to damage the membrane. So here we can see a little bit more of the dystrophin protein. So it has two ends. At one end, it binds to, as I said here, the inside the cell actin, and the other end binds to the membrane. The in-between part is actually made of these blue colored repeat many times. And it's like a spring. It can give flexibility to the protein. So I will talk about micro and mini dystrophin, which is being used for gene therapy. So there we can't use such a long protein because too long. So they have made it short. To make it short, these repeat part has been reduced because the two ends are very important. But in between, if you remove the repeats, then the protein can still function, but it becomes a little rigid. That flexibility of spring is not there so much. Then here I am showing that this is a very long gene, and the gene is composed of exons and introns. So I will not go into detail of this, but this DMD gene has 79 exons. Here I just want to point out this thing, that the way the exons 1, 2, 3, they join to each other, that is very important, because if they don't join correctly, then the protein will not make any sense. That means when the messenger RNA is getting translated into protein, the sense will be lost. I will explain that also, but here you can just see that, like say exon 1. Exon 1 is fitting nicely with 2 because one has a arrow outward and 2 has an arrow inward, so they fit very nicely. But suppose 1 was asked to join 3. 3 is flat over here, so it will not fit properly, and that will create problem in reading rest of the messenger RNA. So that is something that is a problem in DMD. I will explain that. Now, what are the types of mutations that we see commonly in the DMD gene in patients? So we have large deletions and large duplications. About 80% of the time, these are the mutations that we see. And these deletions and duplication, they change what is called the reading frame. So as I was saying that if the exons don't join properly, then the protein is not read correctly. So that if the reading frame changes, it doesn't make any sense. So 80% of these mutations actually lead to changes which can destroy the reading frame. And 10% are the nonsense mutation. That means stop stop signal was put in a place where it should not be stopped. So it stops, and now the protein is not made fully. So exon skipping is a technology that can help in this situation. So let us see how. So, just a minute. So here I've just given an example. See, the way in which we read messenger RNA into protein is something like the English language, you can say, or any language is made of alphabets. And if we put together these alphabets, then how do you read it? So like I have written something here. You can't read it unless I tell you the rule. Now, if I tell you that the rule is that after every third letter, it's a different word. So then you can read that it means you are the boy who won. So this is a normal situation. If this is the messenger RNA, then this is the protein. It says you are the boy who won. But let's say that we have a missense mutation. That means instead of this A, it has now become B. So what will it read like? You breathe the boy who won. So some sense is lost because what is you breathe the boy? But at least some sense is also retained in missense mutation. Now the problem is in this frame shift where the reading frame is gone. What happens here? Here now let's see that let's say that there was we removed two letters. A and R was removed. Now you start reading it. So U is okay. After that, the reading frame, it shifts because the rule is that you have to read 3 at a time. So AR is gone. So after U, we have ETH, then EBO, then YWH. So US above something, it makes no sense. So that's why when the frame shifts, then the whole sense of the rest of the protein is gone. Now, in the nonsense mutation, so let's say that here at T, a nonsense mutation happened. So it should not have stopped here, but it stops. So you make only UR. Rest of it is not made. So these situations can be helped by exon skipping. So how we can do it? So we, for example, where the AR was deleted and the frame was lost. So after AR, suppose I also remove ETHE. Now I have made a little bit more deletion. So now I'll say you boy who won. So little bit a part is gone. Instead of you are the boy who won, I'm saying you boy who won. But at least some sense is restored and the rest of the protein can be made. So that's why exon skipping is a very powerful tool in which we can remove the part which is causing a problem. It can be deletion or it can be the nonsense mutation. It can be removed and the exon can be now linked to the next exon, which makes sense. The frame is restored. So now you can keep reading the protein. So that is the theory of exon skipping. So here we can see that exon 45 has been deleted in some patients. So normally it should be 43, 44, 45, 46. But because of the deletion, it is 43, 44, and then 44 is linked to 46. But this compatibility is not there. So the frame is lost. Now what they do, 43 actually is compatible with 46 because the shape is matching. So what they do is that they remove 44. So in 44, by exon skipping, that is also removed. 45 was already removed because of the mutation. Now they remove 44 also. So now 43 joins to 46. And this is in frame. And now rest of the protein can be made. So, so now I'll tell you about the various ways by which people are trying to tackle the problem of DMD at various steps. So like here, since we already talked about exon skipping, so I'll tell you about I'm only telling very briefly, and I'm telling those where already some products are available or they are in clinical trial like that. So we have nonsense suppression. That means if the nonsense codon has wrongly come, then how to suppress it? So this PTC Therapeutics has come up with a drug, ethyl which is a small molecule and it can suppress the nonsense codon. So even if nonsense codon has come, but it will not stop and the protein will continue to be made. So this drug is actually there, but the problem is that its efficacy is not that good. So they actually showed in a trial that the ambulation was significantly improved in patients for about 3.5 years. Those patients who were taking it were longer ambulation. And there was also lung function retained. So in the European Union, they have also got license for it, but in the United States FDA, it has not been approved because they say that it's you have to improve it. It's not sufficient. Whatever improvement is being seen. So advantage of this one is that it's an oral drug. So you can take it at home itself. But disadvantage that efficacy is very minimal, and it will also benefit only 10% patients where the problem is because of the stop codon. The others will not be benefited. This is about the nonsense suppression. Then exon skipping, I'm sure all of you have heard a lot about it because we have these antisense oligos which are used for exon skipping. And there are several companies in the market, but Sarepta is the one which is leading, and they've come up with quite a few drugs. So like for different exons. So they have for exon 51, exon 53, exon 45. So once you know how to do it, then it is a matter of making specific antisense oligo for the exon that you want to skip. So they have the technology, so they are able to make for all exons where skipping is needed. But the cost is very high and it is weekly intravenous injection. So that's quite a problem, because the child has to go every week for intravenous injection. The other thing is that although it is working, but the efficiency is still not of that level. So Sarepta is working on it, they're improving it, and they're using a different technology with peptide conjugated PMO, which they have shown that it can penetrate into muscle, heart, and diaphragm at better efficiency. So they are doing clinical trial for this. So the advantage of exon skipping is that it is more effective. 7-fold increase was seen in dystrophin levels, and lung function is also better. But the cost is very high, and it needs intravenous infusion. So what I'm saying is that I'll be telling you about so many technologies. All of them have promise. All of them have limitations. So it is research is going on continuously to improve and get rid of the limitations. But we have to understand that it's a dynamic process, and the drugs are not that, okay, this is the final drug. It's there and it is having some effect, but it is still to be improved. Then gene therapy. So gene therapy, so the dystrophin gene is a very, very long gene. And the vector system that is used most popularly is the AAV virus vector. We need this virus to deliver the gene into the muscle cell. So this AAV vector cannot accommodate such a long gene. So therefore, the gene is being shortened. So microdystrophin or mini dystrophin is being used, which will have some function left of dystrophin, but some function will be lost. So various companies are, they're trying this therapy out. Sarepta again is leading in this. The Solid Biosciences and Pfizer, they're all having their trials for AAV dependent gene delivery. The advantage of this is that it's a one-time treatment. So the child doesn't have to go all the time to the hospital. Once you go and get the AAV treatment, and after that, no need for repeated drug infusion. Also, the AAV vector is quite safe. So the chance of any other problem coming is not very high. But the disadvantage is that very, very high production cost, whether we can afford it even when the technology comes. And most people have already exposed to AAV because it is there, it's a natural virus, it's there in the atmosphere, and we breathe it. So we have antibodies against it. So immune suppression has to be done. Otherwise, it will not at all be effective. And that's why it is a one-time treatment, because after you are given once, second time, if you take it, then your body will react and it will be of no use. So in one go, you have to understand how much virus to give. It should not be too much, it should not be too less, because you can't repeat it. Now, problem is that it can cause liver toxicity. So you can't give too high a dose. And it will not make the full dystrophin, only a part of the dystrophin. But in spite of these disadvantages, it does have the advantage that you can deliver the gene, and the gene can actually correct the function which was lost in the muscle.